So why dance? You'll figure that out very soon, but this is about biosimilars. And to be a biosimilar, you must first be a biologic. So I would start off by essentially describing what a biologic is, and maybe I could clarify as to why it is been such a difficult procedure into finding a proper pathway for follow-on, what would be the equivalent of generics, but would be biosimilars. A biologic drug product is actually made in a living organism, whether it's a cell or something. It has to be in a living cell. And it's, since it is manufactured and derived from a living cell, it has its own complexities. And the complexities go beyond just its chemical structure. Now, and that is the first problem of the whole issue, that unlike small molecules, which we are so familiar with, and we'll go on the next slide as to what the degree of complexity is and how much higher it is for a biologic. Just to get the language done, the really key thing is a biologic application is known as a BLA. It's a biologics license application, and very similar to the Hatch-Waxman procedure, the biosimilars come in as an abbreviated BLA, or ABLA. So the typical biologic is a recombinant protein or a therapeutic antibody. And here is the first example of how complex this is. On the left is atorvastatin, known as Lipitor worldwide. In that structure, you can pretty much see the positioning of each and every atom that exists in that molecule. It's drawn in a two-dimensional structure. At most, it would be a three-dimensional structure. There would be certain angles that come out or back into the plane. But it's pretty much exists in only one three-dimensional structure with one chemical composition. On the right is a typical biologic, that a single chain, a single molecule of an antibody, immunoglobulin G. As you can see there, it's in the next slide, I'll show you all the interactions that did happen. It started off with a string of, and let me just go there and show you that. So at the bottom is a primary structure, which is a series of amino acids. So you can think of each amino acid as would be chemically equivalent to a small molecule. Now there's a chain of hundreds of them. Simply based on what amino acids lie next to what other amino acids, it can by itself fold into two kinds of structures, alpha helix or beta sheet, which is a secondary structure on the right of your screen. So after the linear chain has folded along the chain, then it will fold on itself to form a tertiary structure, which is usually the position that biologic takes. And for an immunoglobulin, there is often more than one of these proteins come together to form one immunoglobulin entity, and that would be a quaternary structure. So, of course, the complexity of structures in 3 dimensions is extremely complex. And just going back to the previous slide, there's another level of things that go on. Somewhere in the middle, a little below, is a greenish line. You can see carbohydrates. So sugar residues like mannose, galactose, N-acetyl glucosamine, all these sugars get attached. They get attached based on how and where you grow or make the bacteria. So you make it in an insect or a microorganism or an animal or a human, it will be different. And that will affect whether this molecule is active or not. So bottom line is, for a biologic to be a biologic, not only does it have to have a specific sequence, it also has to be produced in a cell where it is allowed to fold to the correct 3-dimensional structure, and then allowed to associate with the other proteins that have also folded correctly, and be modified with the correct sugar residues. So it's beyond now the structure of pure chemistry. A whole lot of other factors have come in. To give you an example of what this means, on the left is what it would be if I wrote a immunoglobulin molecule down in a linear fashion. On the right is what it ends up looking like. The second part is a lot of this biologic molecule, which you see up here for an immunoglobulin, is complicated, but it's, in terms of functionality purely, it may be 95% replaceable. What do I mean by that? A job of an immunoglobulin molecule is to bind to an antigen, for example, a virus or something like that. That antigen binding part is very small. It's in the variable regions of the light and heavy chains. Those are very small regions with like little yellow dots and things like that. For example, you can make an antibody in a mouse, take out just the antigen binding sites, and transfer it to a human antibody, and it will be functional. But now, what worked in a mouse will work in a human. Why you have essentially replaced 90% of the structure of this, it's still functional against the same antigen in a different organism. So with all these factors coming in, clearly if the patent regime was simply to be able to patent a chemical structure, it won't work, because all these other factors are built into being a biologic. Now, that being said, a biologic can essentially functions most of the time as used as a drug. And if it's used as a drug, then you can, under the FDA, they would also want what would be the equivalent of a generic to come in. And so the biologic application has said the original biologic approved would be known as a BLA, a biological license. And then the first biosimilar would be another biologic that does the same thing that would come in as an ABLA. There are two categories of biologic drug products which can qualify to become a replacement for an approved biologic, which I'll refer to from now on as the reference product. They're biosimilars and interchangeables. The whole intent of how this statute is put together, for you, you'll find it very similar to the Hatch-Waxman rules for drugs. But what has ended up as, is according to the Federal Circuit, quoting Winston Churchill, it's a riddle wrapped in a mystery inside an enigma. Nobody seems to be able to make sense of it, and that will probably be the conclusion you'll arrive at until Jay takes over and kind of explains all the legal battles that have been going on. Okay, so biosimilars are something that's highly similar in that it has the same biological or therapeutic activity. It has the same very similar safety, purity profile. Interchangeable goes a level further. And essentially the minimum definition of the interchangeable is you don't need to talk to a doctor to switch from a biologic to an interchangeable. Think about an original drug, an NDA drug, a new drug, and say a Lipitor for atorvastatin, and then any generic of that drug. You don't necessarily need to consult a doctor to replace one drug with the other. That would be an interchangeable. And of course, the reference product I mentioned is the first in line. So a biosimilar essentially is something, a biologic, that could be prescribed for the same purpose as the reference product was. An interchangeable has a higher standard. In the last section, I say it has to be a biosimilar first. Then it has to produce the same clinical result in any given person. So it should act exactly like the reference product acted. There's no adjustment of dosages or anything needed. While a biosimilar merely has to have the same effect, the interchangeable should be interchangeable. And again, in terms of safety and efficacy, the interchangeable must be at or below, well, more effective, less, and more safe than than the reference product. So let's see what the first thing that was done. So patents are difficult, and we'll discuss it next. But first, just being a biosimilar is being protected by simply giving out directly a market exclusivity, which means the reference product, after its approval, will get 12 years of market exclusivity, which means a biosimilar will not be able to enter the market for 12 years. And also gets 4 years of data exclusivity in which a biosimilar applicant, who has made a biologic that is intended to be something like a generic sort of replacement for the reference product, will not be accepted for 4 years. There are some other exclusivities. They get simply added on. Pediatric exclusivity is simply 6 months longer than any other exclusivities that you got. Marketing, data, and orphan drug. And orphan drug exclusivity, which is for rare diseases, it actually is complex because it runs parallel to the marketing exclusivity. So you have 12 years for marketing, 7 years for orphan drug, and whichever lasts longer is the period you get. So if your orphan drug 7 years expired before marketing exclusivity expires, then you don't get anything from there. So what does the reference product get? No, sorry, let's first do what that was a reference product. Now if you are an interchangeable, which is the higher standard for biosimilar. You have biosimilars, and beyond biosimilars are interchangeables. The interchangeable product, just like the first generic approved drug will get an exclusivity in the small molecule scenario, in the biologic scenario, the first interchangeable product will get a 1-year exclusivity. And it's 18 months to even 42 months, depending on whether a lawsuit was filed that delayed the introduction of that interchangeable product. In contrast, the first biosimilar does not have any advantage over second or third or fourth or fifth biosimilars. They come in whenever they can. Now, how do you decide what's a biosimilar and what's an interchangeable? There are 4 categories that have been defined according to clinical pharmacology guidance provided by the FDA. The first one is not similar, and not similar is not similar. You cannot enter the biosimilar pathway. Similar means there are differences, but if the differences are not significant enough in terms of efficacy and function and safety, then additional studies or modifications can be suggested by the FDA to reach the biosimilar level. Now, highly similar. Highly similar means all the data that is there makes it there's a high level of confidence in approving that drug as a replacement biologic, which again, any uncertainty can be removed with additional studies, but you almost start off with the assumption that it could be replacing. And then there's a high standard of highly similar with fingerprint-like similarity, which means it is essentially very, very close to the original drug in structure, function, everything. And this is about the only case where one could assume that reference for a composition patent on a reference product may be infringed, at least under obviousness standards, by interchangeable, like an interchangeable biosimilar coming in. And that is one little point to note. But other than that, as you've noted all this so far, it's still an exclusivity that's not related to IP. So now we have the purple book. We're all used to the orange book. Now you have a purple book and the purple book was being published from 2014. It publishes the licensing of the reference product as well as approvals of the biosimilar applicants. Now, what's the difference between a purple book and an orange book? Kind of important. The orange book lists patents. I'm sorry, there's a typo there. It lists composition and use patents, the use of the composition patents covering the drug. but the Orange Book does not list method of making patents. It is not that critical when you're dealing with the chemical structure of a small molecule, because you pretty much know what you want to arrive at. And you will, even if you two different pathways, you might still, sets of reactions, you might still get the same end product. The Purple Book, on the other hand, does not list any patents. So that's the second part of the Act. The Biosimilars Act steps in to get these patents out. The only difference now is that patents, including method of making patents, can be asserted during the next step, which is a proposed dance between the reference product sponsor and the biosimilar applicant. As I mentioned right in the beginning, that is very critical, because how you make it is almost decides whether a biosimilar biologic will be functional or not, much more than the actual chemical structure. So the patent dance is proposed, which is a set of moves going back and forth. It starts off with once, let me just give you, it basically is called a dance because there are very fixed time periods in which each party has to respond. Much like a dance, you have timed steps in which each person dancing as a couple has to make a move, and then the other person makes a move, and then you make a move, and so on and so forth. I'm not going to go through this dance, it's boring. But the critical stage is 20 days after FDA accepts a biosimilar application. Somebody makes a biologic, applies for biosimilar approval. Within 20 days, the reference product manufacturer, which would be equivalent of a non-generic maker, the first approved drug in that area, biologic in that area, they need to be given the application that was filed with the FDA, as well as method of manufacture being practiced by the biologic maker. And the whole process is intended to elicit the patents that go. So essentially then the reference product maker, we have these patents, and the biologics maker says, the biosimilar applicant says, well, we may infringe these patents, we don't infringe those patents, and then you come with a list of patents to fight it out over in the end. Oh, sorry, I went the wrong way. Now, you can cut out of all this. You can refuse to dance. You just say, forget it, I am not going to dance. So then you have a second way of dealing with it. The applicant has to provide a 180-day notice of when it will start marketing, commercial launch. Within that 180 days, the reference product maker can sue on whatever patents it thinks the biosimilar applicant has infringed. And this could include any patents, including the method of manufacture patents. It may or may not know, and it could find out how it's done during the discovery at the litigation stage. So, and again, very similar to the Hatch-Waxman, filing a biosimilar application is considered an act of infringement. And there's one little tweak to this statute which I would like to bring out, and that is, it is an act of infringement if the purpose is to obtain approval for the commercial manufacture, use, or sale of a product claimed in a patent, etc. The scope of the patents is any patent for which infringement could reasonably be asserted. Okay, that's fine. Now you can add the method of manufacture patents, and like Hatch-Waxman, but there's a purpose clause in there. And does the purpose clause have a limitation to what kind of patents you could assert? Because it says manufacture, use, or sale, so the composition, method of use are there, but method of manufacture may or may not quite be there, so there's some controversy as to whether the purpose clause affects the scope of what you can sue on. Not been decided yet, not interpreted yet. There are 8, 2 more there, so far pending biosimilar patent litigations. based on being on various stages of this dance or don't dance scenario that was suggested. So far, everything that has been applied for is a biosimilar. No interchangeables have come into the picture. I would think it would be very difficult technically to come up with an interchangeable, but simply a biosimilar is a lot lower burden because you have the same effect with changes in whatever doses or methods of manufacture, etc. Most of the litigation issues I'll leave to Jay to later on to elaborate on, but there was some, there were two things that have been kind of decided at the Federal Circuit level. It kind of plays into this regulatory picture. I just give you the answers, not the reasons. One is, can a company which is planning to file an abbreviated BLA, can it go and sue for declaratory judgment to invalidate the reference product maker's patents?